Chapter 17, Segment 4, pages 559 through 570. Synaptic plasticity can be measured in simple hippocampal circuits. Donald Hebb, 1949, proposed that when a presynaptic neuron continuously activates a postsynaptic neuron, the synaptic connection between the two becomes stronger and more stable. Cells that fire together wire together. Cell assemblies are ensembles of neurons. Hebbian synapses are cell assemblies that are linked via their synchronized activity, which allow them to act together and store memories. In the 1970s, researchers discovered a form of neuroplasticity that appeared to confirm Hebb's theories about synaptic remodeling. Electrodes were placed into hippocampuses, so scientists can stimulate a group of presynaptic axons and instantly record the electrical response of a group of postsynaptic neurons. When researchers applied a brief high-frequency electrical stimuli, a tetanus, to a presynaptic hippocampal neuron, the response of the postsynaptic neurons changed by producing much larger EPSPs in response to normal levels of presynaptic activity. The synapses appeared to become stronger and more effective. Long-term potentiation, LTP, stable and long-lasting enhancement of synaptic transmission. LTP is probably a feature of all neural systems. LTP lasts for weeks once it is formed, and LTP appears to have all of the signatures of cellular mechanism of memory. Long-term depression is a weakening of synaptic efficiency through a different mechanism and can also encode information. LTP occurs at several sites in the hippocampal formation. Hippocampal formation is the hippocampus and the dentate gyrus, as well as the hippocampal gyrus. There are three major divisions in the hippocampus: CA1, CA2, and CA3. LTP was originally demonstrated in the performant pathway, which is the main input pathway to the hippocampal formation. LTP may be a property of all excitatory synapses. CA1, LTP occurs at synapses that utilize the excitatory neurotransmitter glutamate, and it relies heavily on the NMDA receptor, which is a glutamate receptor subtype. NMDA's selective ligand is N-methyl-D-aspartate. Treatment utilizing drugs that selectively block NMDA receptors prevent new LTP in the CA1 region, but they have no impact on LTP that has already been established. AMPA receptors or postsynaptic NMDA receptors work with these related subtypes of glutamate receptors. NMDA receptors and AMPA receptors collaborate in LTP. The release of glutamate at a CA1 synapse activates only AMPA receptors during normal low-level activity. NMDA receptors can't respond to glutamate because of the magnesium blocking the NMDA receptors' integral calcium ion channels, so only few calcium ions can enter the neuron. When larger amounts of glutamate are released, the AMPA receptors are stimulated more intensely. AMPA receptors emit sodium ions, and when they are activated, and so the increased activation of the receptors depolarizes the postsynaptic membrane. If a threshold of negative 35 millivolts is reached, magnesium plugs are driven away from the central channels of the NMDA receptors, and they are now able to respond to the central channels of the glutamate, as they emit large amounts of calcium into the postsynaptic neuron. NMDA receptors are fully active only when gated by a combo of voltage, polarization via AMPA receptors, and the ligand, glutamate. The large influx of calcium at NMDA receptors activates protein kinases, which are intercellular enzymes that alter or activate various other proteins. CAMK2 is a protein kinase, calcium calmodulin dependent protein kinase 2, that affects AMPA receptors in various ways. Activated CAMK2 causes more AMPA receptors to be produced and inserted into the postsynaptic membrane, and existing nearby AMP receptors are forced to move to active synapse. Membrane-bound AMPA receptors are altered to increase their conductance of sodium-potassium ions, and therefore the net of these alterations is to enhance the sensitivity of the synapse to glutamate. CREB, cAMP-responsive element binding protein. This is a transcription factor. Transcription factor is a protein that binds to a promoter region of genes and causes those changes to change their expression rate. And this is activated by protein kinases, including cAMK2 and its chemical cousins. A direct result of activation of NMDA receptors is the activation of CREB and alterations in genes encoding a wide range of proteins. The affected genes may encode anything from new receptors and kinases to the structural building blocks using for altering the shape of the cell, and so this action may have crucial and long-lasting consequences for the neuron. Long-term changes range from formation of additional synapses and enhancement of existing ones, to the construction of whole new dendritic branches and spines via molecular components of the cell skeleton. Not all changes in LTP are postsynaptic, though. When a postsynaptic cell is intensely stimulated and its NMDA receptors become active and admit calcium, an intracellular process causes the postsynaptic cell to release a retrograde messenger, which is released often as a diffusible gas. It's an intracellular process that occurs when the postsynaptic cell is intensely stimulated, and this results in its NMDA receptors becoming active and admitting calcium. The messenger then travels back across the synapse and alters the functioning of the presynaptic neuron, which ensures that more glutamate will be released into the synapse, and therefore strengthening the synapse. LTP involves active participation on both sides of the synapse. Nitric oxide, carbon monoxide, arachidonic acid, and nerve growth factor are various forms of dozens of possible retrograde signals in LTP. In other locations of the hippocampus, such as mossy fiber pathway, LTP can occur without NMDA receptor activity. Some forms of LTP are blocked by drugs with completely different modes of action, such as opiate antagonists. Is LTP a mechanism of memory formation? LTP isn't only a mechanism of learning. It is thought that LTP is a crucial part of a multifaceted system for storing information. Evidence from various research perspectives implicate LTP in memory: correlational observations, somatic intervention experiments, and behavioral intervention experiments. Correlational observations, or time course of LTP and memory formation, are similar to one another. Both can be induced within seconds, they can last for weeks, and both demonstrate a reliable consolidation period that lasts for several minutes after induction. Somatic intervention experiments are pharmacological treatments that interfere with LTP and how they intend to impair learning. NMDA receptor blockade affects performance in the Morris-Water maze and various other types of memory tests. Many of the same physiological processes are at work throughout the body, which made it difficult for earlier researchers to know exactly where and how the drugs were acting to affect memory. Due to recent advances in technology, researchers now can focus in on specific brain regions via regional genetic manipulations. Drugs that inhibit cAMK2 and other protein kinases also interfere with memory formation. Mice with one copy of the cAMK2 gene knocked out can still form STMs, but not LTMs. Knockout mice that lack functional NMDA receptors only in CA1 receptors appear normal, except their hippocampi are incapable of LTP and their memory is impaired. Mice have been engineered to overexpress NMDA receptors in the hippocampus, and these mice demonstrate a more enhanced LTP and above-average long-term memory. Behavioral intervention experiments, or behavioral LTP. This is a demonstration that training an animal in memory tasks can induce LTP in the brain. This is the most convincing evidence between learning and LTP. This research is difficult because of the question regarding where to insert the recording electrodes to detect LTP. Fear conditioning produces LTP specifically in fear circuits in the amygdala. In the CA1 region of the hippocampus, a different version of aversive learning in rats produces the same electrophysiological changes, as well as changes in the rats produces the same AMPA receptor accumulation that are seen with normally induced LTP. Research findings suggest that LTP is a type of synaptic plasticity that underlies certain types of learning and memory. In the adult brain, newly born neurons may aid learning. Adult neurogenesis. New neurons are produced in the brain of adult mammals, and this occurs primarily in the dentate gyrus of the hippocampal formation. This has also been observed in olfactory bulbs in various cortical regions. New dentate neurons that grow will receive input from entorhinal cortex via the performant pathway, extending their axons and forming the glutamatergic synapses. The new neurons appear to integrate into the functional circuitry of the hippocampus and adjacent cortex, which is previously shown to be involved in forming new memories. Neurogenesis and the survival of young neurons can be enhanced via exercise, experience in enriched environments, or memory test training, as well as reproductive hormones and experience. Newly generated neurons located in the dentate gyrus show more plasticity-enhanced LTP. Demonstrations that these new neurons have crucial functions of behavior, especially memory and learning, have been limited though. Rats were given a drug lethal to newly born neurons and showed impaired results on tests of conditioning, but this was only when there was an interval between the CS and US. This type of conditioning is believed to rely on hippocampal functioning. So this type of finding that it is impaired when neurogenesis is prevented suggests that the role of neurogenesis in memory may be limited to hippocampus-dependent forms of memory. Neurogenesis has also shown ties to other forms of hippocampus-dependent learning, such as spatial memory and fear conditioning. Conditional knockout is a gene that can be selectively deactivated in adulthood in specific tissues. In mice with a conditional knockout, researchers discovered that by turning off neurogenesis in the brains of adults resulted in impairment of spatial learning with little effect on behavior. Learning and memory change as we age. By 2030, there will be more than twice as many senior citizens alive than there were in 2000. Healthy elderly individuals show some impairments in tasks of conscious recollection that require effort and that rely primarily on internal generation of memory rather than external cues. Working memory and the ability to form new episodic and declarative memories decline with age, but existing memories such as autobiographical and semantic knowledge stay relatively intact. When vocabulary is tested in elders in isolation, older adults outperform younger adults. On tests of executive functioning, though, individuals even in their 40s demonstrate a decline in memory due to age. Aging results in a decrease in spatial memory and navigational skills. Age-related impairments of memory have several causes. Impairments of encoding and retrieval, loss of neurons and/or neural connections, and problems with cholinergic neurotransmission. Impairments of encoding and retrieval. Elders show less cortical activation when encoding or retrieval is self-activated. Loss of neurons and/or neural connections. The brain loses weight after the age of 30, and some parts, such as the frontal cortex, lose a larger proportion of volume weight than others. Not all researchers agree on this, but memory impairment due to aging may have something to do with steady loss of synapses and neurons in the hippocampus and cortex. Problems with cholinergic neurotransmission. Two subcortical regions, the septal complex and the nucleus basal of Maynard, NBM, provide copious cholinergic inputs to the hippocampal and cortex. Memory impairment in Alzheimer's seems to be caused by the loss of cholinergic afferents to the cortex. Drug treatments that enhance acetylcholine transmission improve aspects of memory performance in human participants. Can the effects of aging on memory be prevented or alleviated? Nootropics. Drugs that enhance cognitive function. Pharmacological approaches to intervene with the effects of aging on memory involve drugs that inhibit acetylcholinesterase, which prevents the breakdown of acetylcholine. The result is an increase in cholinergic transmission in the forebrain, which has had a positive effect on memory and cognition in mild to moderate cases of Alzheimer's. An example would be Aricept or donepezil. Ampakines are compounds that act via glutamate receptors to improve hippocampal LTP. Currently, this is under study as a potential memory enhancer. Other drugs target the family of protein kinases that potentially may contribute to long-term maintenance of memory traces. Rats raised in rich conditions displayed improved ways of handling stress, which reduced glucocorticoid production, which prevents lifestyle-related hippocampal damage. Physical activity, mental activity, and a sufficient amount of sleep have been shown to prevent cognitive decline in the aging process. Longitudinal studies suggest that living in favorable environmental conditions, being involved in complex and intellectually stimulating activities, and having a significant other with high cognitive status reduce the cognitive decline that comes with age.